Hi, welcome to Neuroverse
a podcast hosted by Clara
and Carolina, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on synapses and the evolution and origin of synapses. So I chose today's topic because of a recent publication that brought back to life an age-old debate between two of the most famous neuroscientists, Santiago Ramon y Cajal and Camillo Golgi, about the basic structure of the nervous system. So back in the 1880s, Camillo Golgi first proposed that the nervous system is a syncytial continuum, so like a web, essentially, of connected cells that are fused together. Whereas Santiago Ramon y Cajal first postulated that the nervous system is actually composed of discrete cells that are connected to each other. And eventually, they together actually came up with the technology to study this and showed that basically Ramon y Cajal was correct. And there was the discovery of these things called synapses or synaptic connections, confirming his theory. So what is a synapse? It's essentially where neurons connect and communicate with one another. And it's where chemical neurotransmission occurs that are then converted to electrical signals. So they are the basis of the nervous system, arguably. And synapses consist of three main components: the presynapse, the postsynapse, and the space in between, which is called the synaptic cleft. So synaptic events are mainly regulated by ions, in particular calcium ions. In the presynapse, neurotransmitters that are packaged in vesicles are released by a mechanism mediated by increase in calcium concentration. And then they get released through the synaptic cleft, which is filled with transsynaptic cell adhesion molecules that regulate a whole bunch of things. And they fuse to the postsynaptic membrane. And the arrival of these neurotransmitters then activates receptors on the membrane, which leads to the flow of a whole bunch of ions that eventually change the membrane voltage and cause an electrical event. So synapses are really key for this conversion of chemical to electrical signals, which is how signals are transmitted in the brain.
And synapses are usually formed between axons and dendrites. So the axon usually is the presynaptic neuron that then connects to the dendrite on the postsynaptic neuron.
Yeah, that's the classical structure of neural circuits, but although that's not always the case. So yeah, that is the most common way to think about neural circuits, but there are also exceptions. As is the case in the brain, it always can be more complex than the simplest example.
Of course.
Another reason why synapses are so key is that they can undergo use-dependent plasticity. So maybe you've heard of synaptic plasticity. These are a set of mechanisms that can change the strength of synapses or lead to the formation of new synapses. They can manifest as short or long-term increases or decreases in synaptic strength. And throughout the past 50 years, scientists have been finding all the mechanisms, the molecular mechanisms that lead to these changes, and all of the receptors and molecular changes that are involved, which I'm not going to go into in this episode. But these changes in synaptic strength, which are known as potentiation or depression, they are thought to underlie the encoding and storage of memories, so that a strengthened series of synapses, which in other words is a strengthened pathway, encodes a memory trace or a memory engram, although it's highly debated as well, and I also won't go into that in this episode.
Mm-hmm. Yeah, too many large topics to cover in a single episode.
Yeah. Okay, so while the synapse is a very highly studied cellular site and is obviously essential for all forms of neural transmission and communication and signal propagation and basically everything that underlies how the brain codes information, little is actually known about the origins of synapses and the evolution of synapses. And of course, as we've spoken about previously on Neuroverse, evolution is always an awesome way to look at the brain and its functions and how the structures are related to the functions. So there was an excellent review on this written by Thomas Ryan and Seth Grant in 2009, and they basically stated a set of questions, which I think are really key. So I'm just going to read them out loud. Firstly, when did synapses first form? What was the identity of the organism that necessitated this evolutionary step? Were the first synapses capable of plasticity? And how are the origins of neurons and synapses related? And then they attempted to answer these questions. So something that's really interesting is that components of synapses, which, as I mentioned before, are really rich in ions, receptors, channels, and a whole host of proteins, these synaptic components actually existed before the appearance of synapses themselves. So this has been shown by looking at some of the most evolutionarily ancient organisms, which are unicellular eukaryotes specifically, which are yeast and amoeba.
Which we've spoken about in a previous episode, and whether or not we question whether amoeba can also be intelligent.
Yes, in our episode on the blob. And it also reminded me of our episode on animal models, and where you spoke about a group that uses yeast to study epilepsy because the same proteins that are thought to be involved in epilepsy are expressed in these yeast.
Yeah,
it's a really similar idea. So in these yeast and amoeba, loss of function studies, so basically removing these synaptic proteins, suggests that these genes and proteins are involved in regulating the cell's responses to the environment, like the cytoskeletal regulation in response to nutrients or pheromones and other chemicals in the environment, but they don't technically have synapses. So that's really interesting because it's a similar function to what synapses do in the bigger picture, but without the existence of synapses.
Yeah, so they're not like clumped together. These genes and proteins are expressed, but they're not composed in a neuron, quote unquote, or in a synaptic cleft.
Exactly. And interestingly, also another example is sponges, which is the phylum of Porifera. So these are multicellular organisms that lack neurons, and they also have been shown to have a number of different channels and receptors that have really crucial roles at synapses, including GABA receptors, potassium channels, metabotropic G-coupled glutamate receptors, which are some of the most common receptors that are activated during synaptic neurotransmission. And the functional implications of these receptors have also been demonstrated, similar to in the yeast. Application of an electrical or a tactile stimulus to a sponge causes it to propagate electrical impulses that control water flow through the organism because they exist in the sea. And also, random interesting side note about this is that they don't contain ionotropic glutamate receptors, which are the glutamate receptors that mediate the fast glutamatergic neurotransmission, which is the excitatory neurotransmission. And this suggests that the sponges may have evolved inhibition before excitation.
Oh, that is so interesting. We'll definitely get back to that.
So overall, without discrete synapses, these evolutionarily ancient organisms still have synaptic proteins that carry out similar functions. But then it was suggested that over time, the synaptic proteins have expanded in a divergent manner because vertebrates, for example, have more key synaptic components than invertebrates. So they have more variety of them. And also, there has been 2 rounds of whole genome duplication within the phylum of chordates, which are animals with a notochord, which includes vertebrates, and invertebrates, which includes mammals and humans. And so this whole genome duplication functions to contribute to divergence. And another key thing is that with a larger variety of proteins, there's also a larger variety of combinations of protein-protein interactions. So that's an added level of combinatorial complexity, because at a synapse, it's all about synaptic interactions that hold the pre and post synapse together.
Yeah, really interesting. I've heard Seth Grant speak a lot about this genome duplication phenomena, and he really argues for how it's the origin point in the increase in complexity, which then later on allows us to have higher cognitive functions, and that, an increase in complexity means an increase also in function. Yeah, it's interesting. And even though I understand the arguments and it makes a lot of sense, is this genome duplication enough to account for our complex neuronal interactions? And is it in this literal of a way? In addition to that, with genome duplication, it's not about simply having more genes, it's about the same genes just being multiplied twice. And in one sense, it allows for redundancy. So if for some reason a gene isn't expressed, the other one may be. And they argue that, well, not only Seth Grant, but also another review by Detlef Arendt, where he says that there may be two things happening, that the genome duplication may allow for new protein variants to result and for more specialization. However, there's also an opposing argument that there may be module integration happening. So module integration is basically where distinct modules become functionally integrated. So like, let's say you have like a series of distinct proteins, and like, for example, with the yeast, they then, instead of it just being floating around, we now have the neuron, which it's all integrated into one component, into one organelle. And this allows for more complex functions. So is it necessarily that our genes are duplicated, or is it that they've assembled and organized into this neuronal unit? Which is why I wanted to question whether is duplication enough? Is this increasing complexity and leading to function structure, or is it not, in this other argument about module integration?
Wow, that's really interesting, because maybe it's both. Like maybe it's the duplication that led to the establishment of neurons. This reminds me of our episode on computational units in the brain, because I remember saying that a potential computational unit is the gene, but it could also be the synapse, but it could also be the neuron, and it could also be all. Yeah, maybe the genome duplication did contribute to the module / computational unit formation, leading to enhanced computation. I don't know the answer though.
No, yeah. No, but yeah, it's interesting to debate, and I guess to question whether it's a simple structure function relationship or if there's something else happening, such as integration of parts.
But I also think the key is with the duplication, like you said, it's not necessarily the number of genes, but the number of copies. So with that, you could also have more mutations. You could have more functional implications of mutations, because a mutation on one copy versus on all copies can be different in terms of the phenotype. So,
and that's exactly what Seth Grant says, in that when you have more genome duplication, you have more room for mutation and therefore variation, which then increases complexity.
Yeah, fascinating. Back to synapses, though. Overall, Thomas Ryan and Seth Grant in 2009, they proposed this synapse first model, is what they called it, which is the hypothesis that the emergence of a synapse was a crucial step for the evolution of neurons. So actually, this is very related to what we were just saying. And they argued it's because a neuron is defined by its synaptic connections. And it is conceivable that synapses evolved before axons or dendrites evolved, because an organism with few closely associated neurons, such as the most ancient multicellular eukaryotes, would not necessarily need axonal projections for these neuron-to-neuron communications, but they would have synapses, or at least synaptic components. And so they argued that without these synaptic specifications, axons or dendrites wouldn't have any relevance. In other words, synapses give neurons their cellular and subcellular identity, and it makes it likely that synapse formation may have also evolved before other stages of neurodevelopment, like neuronal migration and things like that actually happen before synapse formation within an organism's life.
Nice. Yeah. No, that's really interesting about how potentially the synapse and the mechanisms underlying it happen before other neuronal migration events.
Yeah, it's a bit counterintuitive. But then, okay, to contrast this all, let's talk about the recent study that was done on these cool organisms called comb jellies, or ctenophores. So Powell Burkhart and Mikey Kittleman and colleagues studied comb jellies, or ctenophores, which are these gelatinous marine invertebrates that move through the water by their ciliary comb rows. And these are among the earliest branching extant lineages of the animal kingdom. They split away from other groups of animals really early on. And so they wanted to study the nervous system of ctenophores because they hypothesized that this early split indicated that the nervous system could have evolved twice, once with these ctenophores and then another time with all other animals. So ctenophores have these elaborate nervous systems that are unique because they consist of a subepithelial nerve net, also known as a, SNN for short, and other neuronal types that also connect to this nerve net. And in the nerve net, multiple neurites that extend from a single cell body are interconnected through anastomoses and form an extensive continuous network within a single nerve net neuron. But they wanted to look at the structure of the nerve net and its connection to other neuronal types in detail. So they used a high pressure freezing fixation technique with ultrastructural analysis using serial block phase scanning electron microscopy, and then established a 3D network of nerve net neurons from this data.
Interesting. Love a good electron microscopy technique.
Yeah, definitely. It's a significant paper because it's one of the first that have looked at this species, the nervous system of this species, in this much detail with high spatial resolution. So the key finding, which, as I mentioned in the beginning of this episode, brought back to life this debate between Cajal and Golgi, which we thought was settled because Cajal was correct and synapses exist and neurons exist. But this study showed that the nerve net forms a continuous structure with no evidence of synapses. So they had synapses elsewhere in the organism. For example, there were gap junctions, which are electrical synapses, identified within the comb plates, and there were chemical synapses identified between the nerve net and other neuronal types, like the sensory neurons of the ctenophore. But within the nerve net itself, there were no synapses. And when I was reading a news article about this, one of the lead researchers said that they found this extremely surprising. So they actually tried to go back and check, are there really no synapses? And there weren't. So they argued that this supports the hypothesis that indeed the ctenophore nervous system evolved separately to other animals. And obviously, the lack of synapses, though, raises the question of how information is transmitted. So they carried out a genome and transcriptome analysis, which revealed that the nerve net neurons do express voltage-gated calcium, potassium, and sodium channels in a number similar to those in neurons of other animals. So they likely produce electrical events like membrane potentials and action potentials, but it isn't necessarily clear exactly how, since we're so used to the idea of these being transmitted across chemical synapses. Two important things to note, though, is that this study was done at a relatively early developmental stage in the ctenophores. So it's not really comparable to the mammals or animals we're used to studying, as you can imagine, in terms of developmental stages. But an analysis at a later developmental stage may show different results. And they also only analyzed 5 nerve nets out of thousands within this organism. So there's still more work to be done. But of course, I mean, especially for anyone who's done electron microscopy or these sort of studies, it's very time consuming. So it's a start for sure. And I think Camillo Golgi would be happy to hear that at least his theory applies to some organisms.
Yeah, that's super interesting and fascinating how they have, I guess, all of the components to communicate via chemical synapse, but then they don't. But I don't think that, like what you mentioned, the development of ctenophores may have just been different from the development of other eukaryotes. And I think one doesn't exclude the other necessarily. Perhaps it's just a branch in the evolutionary tree that like happened a bit earlier on than we think. And related to that, so there was this review written by Detlev Arendt, which I've mentioned already. It's the Evolutionary Assembly of Neuronal Machinery. And I really recommend as a review. It's super well written and very easy to digest. And he basically breaks down the different components of the neuron and I guess how they came about evolutionarily and what came first. And he breaks down the different components of the synaptic machinery and how it emerged through modifications of calcium-regulated vesicle release in terms of the presynapse, and then in terms of the postsynapse, that it has the glutamatergic postsynapse, which originated through fusion of sensory signaling module and a module for filopodial growth, while GABAergic postsynapse incorporated an ancient actin regulatory molecule.
That makes sense because in general, excitatory synapses mainly form on what we call spines, which are like filopodial outgrowths from dendrites on the postsynaptic side, whereas inhibitory neurons mainly form on shafts.
Yeah. And so he goes on to try and figure out which came first in terms of either the presynapse or the two different components of the postsynapse. So he has a few criteria which he tries to follow to try and figure out this. So he looks at either the degree of structural functional independence in the neural context. Then he also looks at whether or not they exist as autonomous modules in other non-neuronal cell types, like we've spoken about with yeast. And then also individual components, whether they're older than the composite neuronal structure in evolutionary terms. And I think that, like in Seth Grant's paper, we can see he mentions how certain genes that focus on anatomical specification that happened earlier on in the evolutionary tree. So we can date these genes and see when they were introduced or expressed. So following this criteria, there are larger differences in the molecular architecture for the postsynapse, indicating independent evolutionary origin from the presynapse. So he speculates that the excitatory and inhibitory postsynapse arose separately, but building on the same junctional core and incorporating distinct receptors and proteins. So this is related to what you were saying earlier, in that glutamatergic and GABAergic synapses were found on the same dendritic arbor, but there is some debate as to which one occurs first. And I think David Lent argues that evolutionarily, there's more evidence for glutamatergic postsynapse to emerge first. But I guess this is still up to debate. But I really enjoyed the way he broke down the different components, and he goes down to the detail of the different neurotransmitters, glutamate and acetylcholine and GABA. And yeah, it's a really interesting review.
Yeah, sounds interesting. And I guess in contrast to what we said earlier about in terms of the proteins themselves rather than the structures, GABAergic proteins such as receptors were found in evolutionarily older animals before the emergence of the glutamatergic ones. So maybe there's also a dissociation of like the presence of the protein and the presence of the structure and the functional module, as you said.
Yeah.
So I think that's a great place to sum up today's episode. Thank you so much for listening to the episode, and I hope you enjoyed learning about synapses.
Yes, and if you enjoyed this episode, please feel free to share it with your family, friends. It means a lot to us. And check out the website for resources.
Also follow us on Instagram and Twitter at neuroverse_pod. Thank you for listening.